Well, okay. Welcome to 10.4 on cell differentiation. The goal is to analyze strategies for prevention, detection, and treatment of communicable and chronic diseases. This is our last lesson for this unit on cells. I hope you enjoyed all the fun activities and things we've learned about, and all about cells, obviously. So we'll take a look at the far side and enjoy that for a second. Now let's look on to our objectives. To describe the process of differentiation, define stem cells in their importance, identify the possible benefits and issues relating to stem cell research. All right, it's all about stem cells today. So think about it. The human body contains hundreds of different cell types, and every one of them develops from the single cell that starts the process. So how do the cells get to be so different from each other? So it's called the process of differentiation. So all organisms start life as just one cell, right there, called a zygote. Most multicellular organisms pass through an early stage of development called the embryo, which gradually develops into an adult organism. So during development, an organism's cells become more differentiated and specialized for particular functions. For example, a plant has specialized cells in its roots, in its stems, and its leaves. The process by which cells become specialized is known as differentiation. During development, cells differentiate into many different kinds and become specialized to perform certain tasks. Differentiated cells carry out the jobs that multicellular organisms need to stay alive. So in some organisms, a cell's role is determined at a specific point in development. So they determine their role, okay, that's a good word to use, at different times. That's what I'm trying to say. In the worm here, for example, daughter cells, all right, from each cell division follow a specific path toward a role as a particular kind of cell. So you can look here, the nervous system is developed by the 5th cell division, the cuticle is developed by the 8th cell division, and the pharynx is developed after 9 to 11 cell divisions. All right? So cell differentiation in mammals is controlled by a number of interacting factors in the embryo too. Adult cells generally reach a point at which their differentiation is complete, and then they can no longer become other types of cells. So the process of differentiation happens during the development of an organism, and cells differentiate into many different types of cells. A basic overview answer there for you. So objective 2 is define stem cells and explain their importance. One of the most important questions in biology is how all of the specialized differentiated cell types in the body are formed from just a single cell. Stem cells are unspecialized cells from which differentiated cells developed. And there are 2 types of stem cells. There's embryonic and there's adult stem cells. And there are 2 types of different embryonic stem cells. Biologists say that a cell is totipotent and it's literally able to do everything to form all the tissues in the body. So the totipotent cells happen after the egg has been fertilized, and it's only divided a few times. So only the fertilized eggs and the cells produced in the first few divisions of the embryonic development are truly totipotent. After that, they can't become just anything, all right? They can become most anything, but not just anything. So totipotent cells are just after the first few divisions, and they're literally able to do everything to form all the tissues of the body. All right. So after about 4 days of development of the fertilized egg, the human embryo forms what's called a blastocyst. It's a hollow ball of cells with a cluster of cells right here inside, and this cluster of balls of cells is called the inner cell mass. The cells of this inner cell mass are said to be pluripotent, which means they are capable of developing into many, but not all of the body's cell types. Embryonic stem cells are found in the inner cell mass of the early embryo right here, and this is what scientists most frequently would like to use, because it does develop. These are stem cells that can be developed into most anything. All right, adult organisms do contain some types of stem cells, but they are very difficult to harvest. Adult stem cells, if you can find them, are called multipotent. They can produce many types of differentiated cells, but not everything. And adult stem cells, like if you take it from a given organ, if you can find it in your liver or something, then that can only, if you can find it, it can only produce a liver. So they typically produce only the types of cells that are unique to that type of tissue. Okay? You can find some adult stem cells in your bone marrow, which gives it a little more flexibility. But if you want to develop another heart or something, you're going to need to take it from the heart. That's why scientists like to use the embryos, because they're more flexible, shall we say, and they can differentiate in more different things. All right, so stem cells. Define stem cells and explain their importance. The unspecialized cells from which differentiated cells develop are known as stem cells. Why are they important? All right, identify possible benefits and issues related to stem cell research. Well, they're important, but there's also issues too, obviously. Potential benefits. Stem cell research could lead to many new ways to repair the cellular damage that results from heart attack, stroke, and spinal cord injuries. And one example is the approach to revising heart attack damage illustrated here. So the stem cells are filtered from the bone marrow, removed from the stem cells from the bone marrow or removed from the patient's hip. The stem cells are injected in the heart damaged area. The environment of the heart stimulates the injected stem cells to differentiate into new heart muscle cells. So you can repair heart damage here. What is the opposition to stem cell research? Most techniques for harvesting or gathering embryonic stem cells do cause destruction of the embryo. Government funding of embryonic stem cell research is an important political issue, and groups seeking to protect embryos oppose such research and claim it is unethical. Other groups support this research as essential to saving human lives, and so it is viewed as unethical to restrict the research. So it's back and forth, and as we go, there's always going to be some opinions on that. Objective 3 was to identify the possible benefits and issues relating to the stem cell research. Stem cells offer the potential benefit of using undifferentiated cells to repair or replace badly damaged cells and tissues. Human embryonic stem cell research is controversial because the arguments for it and against it both have ethical issues of life and death. So our objectives were to describe the process of differentiation, define stem cells and explain their importance, and identify the possible benefits and issues related to stem cell research. All of these help support our goal, which was to analyze strategies for prevention, detection, and treatment of communicable and chronic diseases. And there is my very cute puppy picture for you today. All right, guys, we're going to have a unit test on the cells, so I hope you're studying for it. Bye.